Welcome to Neurosalience, the OHBM podcast.
Welcome to the Organization for Human Brain Mapping Neurosalience podcast. I'm your host, Peter Banatini. Here, I interview brain scientists and discuss their work as well as the latest advancements and challenges in the field of brain mapping. My guest today is Dr. Michal Ramot, who is a pioneer in the field of real-time neurofeedback fMRI. This method is a unique and powerful kind of fMRI involving real-time feedback of brain activity to the subject towards the goal of enhancing or suppressing activity or connectivity and ultimately changing behavior. Her work has taken real-time neurofeedback fMRI to the next level, embracing operant conditioning to alter measured fMRI network activity independent of the subject's awareness or conscious control. Here we discuss all the types of neurofeedback-based fMRI, focusing mostly on her implicit neurofeedback studies. We discuss the real-time fMRI feedback setup, as well as the potential applications for understanding how the brain reprograms itself, as well as clinical applications. Dr. Ramot received her Bachelor of Science in Mathematics from the Hebew University in Jerusalem in 2004. She went on to receive her Ph.D. from the Interdisciplinary Centre for Neural Computation, working under the guidance of Rafi, Malach and Deouell. She carried out a postdoc at the Department of Neurobiology at the Weizmann Institute of Science, also under Dr. Malach, and did a second postdoc under Dr. Alex Martin in the Laboratory of Brain and Cognition at the National Institute of Mental Health. She's currently a senior scientist in the Department of Brain Sciences and the Roel C. Career Development Chair at the Weizmann Institute of Science in Rehovot, Isreal. This conversation was just fascinating as it covered so many possibilities and questions regarding how fMRI can be used as a means to change the brain, as well as a tool for understanding how the brain changes itself. Enjoy. All right, welcome, Michal Ramot. And actually, hopefully I pronounced your name correctly, Michal Ramot. Michal.
Yeah, that was pretty close. That was good.
Okay. All right. Well, thanks for coming on the podcast, the OHBM Neurosalience podcast. And today we're talking about something really, I think that has been sort of exciting for a while, but then it sort of died down. Now it's coming back. But the more I look into it, it's really exciting to me. And hopefully we convey that to the people listening to this. It's real-time neurofeedback fMRI. So everyone thinks about clinical applications of fMRI, but this is a huge one. It has huge potential for clinical therapy, but also, as hopefully we'll be talking about, and really understanding how the brain changes itself and how and causality in network activation. So just to begin, and also Michal has been a pioneer in this area for a while and has written some very impactful papers, actually sort of pioneering new types of paradigms as well with real-time neurofeedback. So just to begin, so your background is in mathematics, but then you got into neuroscience, which I think is probably one of the best ways to get into neuroscience with a background in either mathematics or physics or engineering. But so what was your start and what made you sort of become more interested in the brain?
So it was actually kind of random, like it just happened that way because of people I met, I think. So when I was doing my undergrad in mathematics, I was also doing at the same time like a co-major in this interdisciplinary program of the humanities. And the people from this program came from all over. So you did like this program, which was like half of your undergrad, and something else. So some of them came from mathematics, some of them came from physics, some of them came from psychology or like any other humanities background. And our TAs also were graduates of this program, so they also came from all different kinds of places. And some of our TAs were in this computational neuroscience program for grad school. So I learned about this neuroscience program from them, and it sounded kind of cool. And then I finished my undergrad, and I didn't know what I wanted to do in life.
Yep.
So I actually took a year off and went to South America.
Oh.
And I was in South America for 8 months, and like traveling all over and hiking and thinking like, what do I want to do with my life? And I was like, oh, but remember those people, they talked about the neuroscience, kind of sounded interesting. Maybe I'll try neuroscience. So I signed up for the neuroscience program. And I actually knew nothing about neuroscience before I began. So I hadn't studied biology since like 7th grade, because I was always in the physics math direction. Yeah. And I knew nothing at all about the brain. But this program was also like an interdisciplinary program, brought people from all over. So they had this really great introductory course to neuroscience. And there I first learned about action potential. I was like, oh, this is amazing. This is so cool. And then I was in love. And then that's it. I've been in neuroscience ever since.
Oh, well, that's a great story for a start. I think it's underrated, taking time off after. I mean, I think that's almost the case of everyone, and a lot of people actually say, right, when they take some time off, they get a good perspective, and then they can dive into what they want to do. And that's South America. That's awesome. So, and then once you got started, so were there any sort of mentors? So who was your mentor when you started graduate school? Was Rafi Malach?
Yeah. So my main PhD mentor was Rafi Malach, who's just an amazing mentor. I think in general, probably the best I could have asked for. So I think my two main mentors in life were Rafi for my PhD and Alex Martin for my postdoc. And both of them, I think, were just such great examples of what a scientist is and what a scientist should be, and how a lab should be run, and like how you should do science on all different levels. Like this, obviously science, but also in terms of how you manage a lab, how you interact with students, how you help them, how you nurture them. So in that sense, it was amazing. And I think what really got me into neuroscience after this initial like epiphany with the action potentials was Rafi is just so enthusiastic about science. So I remember we would go on lab field trips. Like we would go see flowers, and we would bring wine because it's not an age. And and he would get like a little bit tipsy, and then he would still talk about science. So when he was at his most enthusiastic, this is what he really was passionate about. This is what he really, really cared about. And it was just so inspiring to be so in love with what you do.
Yeah.
So I hope to be in that space as well.
Well, that's yeah. And I think actually, right. And it's a rare person. I think that, right. You pick that up, and I think from my interactions with you, I think that you would fill that role really well. But I do think, I think that many people actually, it's the how they carry themselves, how they run their lab, what their passion is, that rubs off on people almost more so than just information that they give them about what to do and whatever. So that's really important. So you started doing research. He's a vision neuroscientist, and so you started doing some sort of research on more, the pure, either like fMRI or otherwise, I think, and then you got into real-time fMRI and real-time neural feedback. So how did that interest get piqued in your brain?
So Rafi was actually kind of transitioning from being really a vision neuroscientist to more a consciousness neuroscientist. So he's been really interested in consciousness the past decade or so. And I was studying spontaneous activity when I was both with fMRI and also with ECOG in epileptic patients. And then in 2011, the Shibata paper came out. Yep. So this is a paper where this is the first implicit neurofeedback paper, I think. And it's an incredible paper. And they show that you can train the brain to learn without telling you what to do, right? It's like the first time I've ever seen implicit neurofeedback. And I saw neurofeedback papers before, but this was so different. it was so interesting. So like all the other neurofeedback papers I'd seen before seemed like they didn't seem to be doing anything really new. And this was doing something really novel. And I just got so excited about this. And Rafi also got really excited about this. And we started talking about it. And also, okay, so when I was a teenager, I read Heinlein and Stranger in a Strange Land.
Yes. Yes.
Okay, so in Stranger in a Strange Land, which is teenager is the last age at which you should read this book. You shouldn't read this book. I tried, I reread it and it's terrible. But as a teenager, it had a deep impact on me. And in this book, one of the main characters was raised on Mars. And the Martians taught him to do what Martians do, which is basically cure themselves. Right? So they go into this meditative state and then they cure themselves of whatever is ailing them. And I read the Shibata paper and I was like, oh, this is kind of like that.
Yeah.
Right? You're kind of training by showing the brain what it's supposed to do, but without actually without explicitly trying to get there.
Yeah.
You're just letting the brain learn where it's supposed to go.
Yeah.
So I thought that this has such tremendous potential for everything, right? Like if you can do this, if you can teach people to see gratings without showing them gratings, then you can do anything. Like maybe you could rewire the brain in any way you want, which I actually think isn't the case. I think there are constraints on what you can and can't do with neurofeedback. But the basic concept just got me so excited about this field.
Yeah.
And then I knew that this is what I want to do.
Yeah. Yeah. And definitely, and I think actually we'll talk a lot more about that. And I totally agree with you. I think that, the early days of real-time feedback were, I think, for instance, like Chris Ducharme's paper got a lot of attention back in 2004 with, pain mitigation, but still that was sort of along the lines of potentially, I mean, I think it's important to actually spell out the differences in the paradigms a little bit more carefully, because I think people picture, when they picture real-time, they picture sort of consciously trying to do something. It's not clear what strategies people use, but do something to change the brain activity as it's fed back to them for a certain network. In this case, for Ducharme's, it was like a pain network. But you're right. I mean, the Shibata paper, though, I think was a huge shift, and that is the subjects didn't even know what the experiment was, and they were looking at these gratings, and with a certain grating, you had this ball this circle that expanded, and that probably gave a little bit of dopamine or some sort of neuromodulation, maybe in the basal structures, the caudate nucleus or putamen, that then went and said, hey, that whatever that was is good that you were doing, and it happened to be that grating,
but it's all spontaneous activity, right? They're not actually seeing anything. It's just you're kind of monitoring spontaneous activity in the brain as it happens. And whenever you see the pattern you want to reinforce, then you give reward.
Yes.
And it's like a basic operant conditioning approach.
Yeah.
Right? If we were doing this with monkeys, we would give them juice, basically.
Yes. Yeah. And that's so powerful because you're right. You're bypassing the conscious process. So it's kind of like, Pavlov, on steroids in some sense. It's sort of like, you're training the brain networks without your conscious processes being in the loop. But anyway, we're potentially getting ahead of ourselves.
Yeah.
But just why don't you trace back? I think that, like, for instance, way back in the 90s, real-time fMRI was possible with, Bob Cox was one of the, among others, sort of developed sort of the ability at least to bring up fMRI images in real time. And that was great. It was sort of exciting. It was a nice, cool tool to show, for demonstrations, but the use of it didn't come around until maybe, and I think Ducharme's paper was one of the major first ones, but there are others. I think you probably know that better than myself, but then right, the field, and actually that paper, just to reemphasize that it generated so much excitement. I remember at the NIH, there was, there's whole RFAs, requests for applications that were put out with real time in mind, and it was exciting. So why don't you describe, just so people know, the basic real-time fMRI or real-time neurofeedback setup, and independent of whether it's maybe describe the conscious feedback versus what you were describing as well. So yeah.
So the basic setup is the same for the two approaches, right? You put your subject in the scanner, and then you're monitoring the brain in real time, and way in advance you define what networks or regions you're interested in, right? And then you can recognize them in real time. So you have maybe this mask, and you can extract the signal from these particular areas in real time, and then you can do all kinds of, and then it diverges with what you do with the signal. So in the more conscious explicit paradigm, the idea behind it is that you now feed back so you're translating the signal from the brain into something that they can perceive, that your participants can perceive. And by translating it for them and making it explicit for them, you're allowing them to explicitly learn how to control it. Right? So say now if you're interested in an activity in the amygdala, then you can look at in real time the activity in the amygdala, and then you can portray this as some kind of bar on the screen, or something like, say, a bar on the screen, right? And the level of the bar indicates the level of the activity, and you're telling them what this represents. So you say this represents activity in the area of your brain that's involved in emotional processing. So if you want it to be higher, then you should, think happy thoughts, or try and remember emotional events. And you give them an explicit strategy for controlling the signal. And then they see the signal go up and down as they do whatever strategies they're doing. And the idea is that they can then learn to better control it, right? That's one setup. The implicit setup is similar technically, right? So you still have this activity from the brain, but now instead of telling them what to do, you're just reinforcing the activity that you want. So you're not translating the signal, you're not portraying it visually in any sense for them. You're abstractly rewarding the occurrence of the brain state you want to reinforce. Yeah. So this gives you way more flexibility, because you don't need to know what this brain state does. Right? So if you want to give them a strategy, you need to know what the strategy is. If you're not giving them a strategy, then you can now reinforce anything, right? So you can look at how activity in the amygdala correlates with activity in a different part of the brain, and only when they co-activate, you give feedback, and you don't know what would make these two areas co-activate. Right? So you have much more flexibility. And the feedback you give should be kind of decoupled from the network you're trying to train, because you don't want it to activate the network in itself.
Right.
Maybe that's a point we should get into a bit later. We should expand on that.
Okay.
But that's the basic setup. So you have the real-time signal from the brain, and you're either showing it to them explicitly, or you're just rewarding them whenever the activity pattern fits whatever it is you're trying to reinforce.
Yes. Okay. All right. So now that that's clear, I think that, the first thing that was made that possible was, echoplanar imaging, but then also, high-speed processing to actually create these functional images on the fly. And obviously the feeding back is pretty straightforward. But one thing that has always, not really always, but still does, I guess, trouble me a little bit is the hemodynamic delay. So you think with any sort of conditioning, you want to sort of have the time frame of the feedback with whatever you want sort of coupled in time. But the hemodynamic delay is about 7 to 10 seconds, and then the processing, that's probably almost instantaneous. So, but is that delay a significant issue, do you think, or things are not so transient, and hopefully they're spread out a little bit over in time, that it's sort of overlapping with the reward?
Yeah. So I think one of the really surprising things about real-time fMRI neurofeedback is that it works because of this hemodynamic delay, because I agree that you would expect this delay to ruin the effect, to be too large, but it doesn't. So people can still learn even with this delay. So the question is why? And it's possible that it's just that things are, as you said, they're more spread out. So we know the spontaneous activity is slow, right? You have waves that carry on for tens of seconds. So it's possible that you're just in this state for 10, 20 seconds, and that's what you're reinforcing, and then it's fine, even though there's a delay. It's also possible that it is more transient than that, but because the delay is fixed, that the brain can still learn. So it learns to assign that credit to whatever happened a few seconds before, right? Because the delay is fixed, or it could be a combination of the two. It's a good question, but I do think that if we didn't have the delay, so for instance, if you were doing neurofeedback on electrophysiology, I would expect it to work better, because it should be easier to learn without the delay.
Yeah. And maybe just to bring that up really quickly, I think that is there a potential for, I know that people can do simultaneous EEG and fMRI, and I know some groups have been working on that for real-time neurofeedback. Do you see that as being synergistic or somehow helpful to combine the two signals in some way?
Yeah, so I think it depends on what you're trying to do, right? So for clinical applications, definitely, for many reasons. For basic science research, I think maybe not quite yet, because for basic science research, if what you're trying to ask is about very specific brain states, you might not be able to get that level of resolution with EEG. For clinical research, I think that's definitely the direction it needs to go in.
Okay. And what about, I think there's also the question of the fidelity of the signal. So, with fMRI, it seems that you have a pretty high fidelity signal that you can actually use within, a few seconds. But, EEG maybe is a little bit more noisy and it takes a little bit more. So it might be a wash in some sense, but who knows? That's
yeah. Although there are, like you said, there are groups that have been working on this and they've had some success. So like Talma Hendler has been working on this for a long time, and Yeji Budurka was working on this as well. So yeah, I know it seems like you can. There are things you can do with this.
Yeah. Okay. And all right. So So you mentioned the Shibata paper, and I think it's worth sort of mentioning how cool that is. Like you said, they first did an experiment where they did Gabor patches and showed these networks, but then with that data, they did multivoxel sort of pattern effect analysis of this, and then had no stimulus, had nothing. The subject was lying there, and they were given sort of a reward in some sense when that activity pattern corresponding to one of the grading orientations came up. And they did this, and this is an interesting question too, it took like about 10 trials to sort of learn it. And then the amazing thing, and we can talk about this, is that their performance in recognizing that grading improved, and it didn't improve for the other ones. So why don't we talk about those two things? One, how long it takes for this to actually have an effect, and the difference between actually changing the networks versus changing behavior in the subject.
So how long it takes to have an effect, I think, depends on your design in many ways. So for instance, in what they did, because it was a block design, then you have a limited number of trials per run, right? So like each block takes about at least 30, 40 seconds. So, and you get one feedback per block. So in a 10-minute run, you would only have, I don't know how many events, like maybe 15 events. So I think that's why they need lots of runs in order to get an effect, because you need to have enough feedback events for the brain to learn. That's why I was trying to design more continuous designs where you could do a kind of correlation, and you could do it TR by TR. So at each time point, you could give or not give a reward, and that way you could have as many as 200 rewards per 10-minute run, right? So, and we actually show that you can learn within 4 sessions, even less actually, but within 4 sessions, almost all our subjects were learning. Yeah. So I think it's like this balance between having a reward that's accurate enough, so you can estimate the signal accurately enough on the one hand, but also having enough trials. Yeah. So that's in terms of how long it takes. Yeah. And the other question is really important. So, okay, so now you've changed the brain somehow, and what has it done? Right, that's the real question of neurofeedback. So this is what I'm mostly interested in from a more basic science perspective, right? So one of the difficult things, definitely with humans and fMRI, but actually also with animals and any kind of brain research, is getting at the causality. So you have all these correlations between a particular brain state or a particular network, or a particular activation pattern and a behavior, but you don't know if that brain pattern or correlation or network is causing the behavior. And what you want to be able to do is to perturb the brain directly and then see how that changes behavior. And in humans, if it also has to be non-invasive, then you're very limited in what you can do. Yeah. So that's what I think neurofeedback is perfect for. It's like a way of perturbing the brain, because now you can take a brain state and by reinforcing it, you're changing it, right? You're making it stronger, for instance, or weaker. Yeah. And you're doing it in a way that completely bypasses any overt behavior. Yes. And if after doing that, you see a change in behavior, then that really shows causality. That really shows that you had this network and then you magically changed it, right?
Yeah.
And now the behavior is different, then that network controls that behavior.
Yeah, and it seems that it's in this method seems like it sort of makes use of this intrinsic sort of evolutionary system anyway. You have dopamine or whatever reward with behavior, and certainly there's a network in between, but here you go, reward with network, and then it affects behavior. And it seems more natural and potentially more powerful than, obviously there's many neuromodulation methods like TMS or whatever, and that seems somehow unnatural in some sense, where the networks may not naturally be firing in sync or out of sync or suppressed or enhanced. And here it's sort of like you take a network that spontaneously emerges or not, and then you try to
you just reinforce it.
Yeah, just reinforce it and nudge it in different directions. Yeah.
So all the other neurostimulation techniques, for instance, and they're all kind of imposing external activity patterns on the brain, right? So you're kind of hitting the brain with this large hammer, and then it changes, but you don't know exactly what happened in between, right? Because you created all these patterns that never existed. Right. And here, all you're doing is reinforcing existing spontaneous activity. So in that sense, it's much more natural, right? And I think it's a good way of learning about the brain. And the other thing is that you can do this to almost everything. I mean, we did this on correlations, you can do this on activity patterns, but you can also think about doing this on, frequency bands, or if you could measure neuromodulation, specific neuromodulators in real time, you could reinforce, the concentration of a particular neuromodulator. You could do anything, like you could reinforce anything that you can measure in real time.
Yeah. And that is so cool. That is so cool. And you can imagine, right? I mean, it seems like, and from the early work, it does seem like it works. I mean, you have a paper, and we'll talk a little bit about your recent one, but you have one. Why don't you talk a little bit about your paper with the autistic spectrum, and actually you have subjects who had autism, and you essentially, to some degree, change their behavior. And so why don't you mention that a little bit?
Okay.
Yeah.
So the idea here was to really look at whether we could change correlations in a network, because it's like you said, it's not easy to target correlations with fMRI because the signal is slow. And so we had to come up with a way of measuring a kind of proxy for correlations in real time that we could reinforce. And we wanted to ask two things, like, can you change correlations in a network with real-time fMRI? And can you do this in a network that's somehow aberrant, like developmentally aberrant? Yeah. And we know a lot about aberrant connectivity in autism spectrum disorder. And from Alex's lab, there have been lots of studies looking at this kind of aberrant connectivity. So we took a data set that Steve Gotts collected in the lab, and we found the two nodes that were most underconnected in autism compared with controls. And not only were they underconnected compared with controls, but they were also correlated to symptom severity, right? So the more undercorrelated they are, the worse the social symptoms in autism were. Yep. And the question was, okay, so if we now strengthen these connections to make them, these nodes, right, to make them more like the typically developing brain, then will it work? Like, can we do it with neurofeedback? And will it change behavior? Will they become less socially impaired? So we tried this, and we brought them in for 4 different sessions, like they came in 4 times. And it was only about half an hour of training each day. And we just reinforced these connections whenever they co-activated, like these two nodes.
And the way you reinforce it, once again, it was sort of like a hidden picture. And whenever the network, well, you might.
So we thought a lot about what to do, like what the reinforcement should be, because like I said, so we haven't really gone to this yet, and I think we should talk about this more, but we wanted the feedback to be orthogonal to the network that we're training, because you want to completely, if you want to look at causality, you want to completely decouple everything. So you're only really directly changing the network and not doing other stuff that might also affect it, right? So we wanted the feedback to be not related to this network, which again, we didn't actually know exactly what it does. It was STS, part of STS and part of somatosensory. So it's not like I can tell you what to do to make STS and somatosensory co-activate, but because it was related to social symptoms, then we didn't want the feedback to be social in any way. So you want feedback that's kind of orthogonal to social networks, but you want it to be rewarding, because we don't know how it works, but it has to work through the reward system. Right? Whatever neurofeedback is doing is modulated by the reward system. So it has to be rewarding. and it's slow because it's fMRI.
Yep.
So like there's a limit. You can't, like, it would be good to have like a really cool game that they play, but it's super slow because the feedback can only be once every 2 seconds.
Yeah.
So then we came up with this puzzle task game where they start off with a blank screen, and we tell them there's a picture hidden beneath the screen, and your job is to reveal the picture. And good luck. And they're like, what do you want from us? What are we supposed to do? And it's like, oh, you'll figure it out, just try. So then we put them in the scanner and they see this blank screen, and then they come up with all these strategies. But what's really going on is that we're looking at these 2 areas, and whenever they co-activate and not with the 3rd area, then we reveal a piece of the picture, and it comes up with this like happy da-dum sound, right? So you have like that happy sound, and you reveal a part of the picture, and then they get to see the whole picture, and we do this for like 5 different runs, and they get their score at the end of each run, which is like how many squares they revealed. And if they next one they reveal more squares, and they get a bonus, and they get really into this. So it was good in the sense that it felt like it really was rewarding for them. They were really into this game, and that's the important part. Like, it just has to be rewarding.
Yes. Yeah. And so okay, so then so they do this, and they do this several times, and so what
so then what we saw is that, so we were training this like mini network. So we were training these 2 nodes to correlate more and to correlate less with the 3rd control node. And we saw that that's exactly what happened over the 4 days of training.
Yeah.
The correlation between the 2 targets increased day to day, and the correlation between the target and control went down, and then it stayed down. And then we brought them back a month or a year, between a month to a year following the training. And we saw that most of the gains were maintained. So the network was still almost at the end state of the training, almost like even a year after. The other thing was that it seemed to be very spatially specific. So we also did the whole brain analysis where we looked at what changed in the brain following the training. And we saw that what reliably changed was like these areas. So if you ask across the entire brain, which area is now more connected to target 1, for instance, and less connected to control, then you get to target 2. But if you look, so like at the very highest threshold, you get target 2. But if you take the threshold down a little bit, you see that's not only target 2 that changed, but also the entire network that's correlated to it, that's connected to it. So it's like you're changing. And when you think about it, of course, that's true, because you can't change like a single neuron in isolation or a single region in isolation. So everything that happens downstream and upstream is also changing to support it. But the peak of the change was exactly where we targeted. So that was very cool.
That's incredible. And it translated into behavioral measures as well.
Yeah, so we saw correlation between the change in the brain and the change in behavior. It was a bit strange though. It wasn't like they didn't overall get better.
Okay.
But it was correlated. I don't know, I think, so 2 things about the behavior. I think the behavior we measure in ASD, it's a very complex measure, and it's not, it's not a good measure. It's like these parent questionnaires, and they're not meant for this time scale of change anyway.
Right.
So it's amazing that we saw anything at all. Yeah. And it's a very complicated behavior as well. We were only targeting like a part of this network. So we know there are lots of different networks that are aberrant in ASD. Yeah,
and it might be right. It would be interesting to actually fully characterize the networks and maybe go through and sort of change each one of them. And but I mean, there have been other examples of I think Michelle Hampson at Yale has been sort of pioneering this as well. And she actually found some, without going into her paper that much, that with clinical symptoms that that subjects not only got better, but then once they were sort of in this different trajectory, they kept on getting better after the therapy, which is interesting in itself as well.
Yeah, so that really fits with the way I'm thinking about neurofeedback, about how it can actually work. Because like I said, we brought them in for half an hour of training 4 times. So it's like 2 hours of training of their entire lives. And it had a significant effect on the networks. And you're like, how is that possible? How can you change networks in 2 hours of this noisy training with the HRF delay and all that? And I think this happens because the networks are already in this kind of suboptimal state. And what the training is doing is kind of pushing them out of this local minimum, and it's bringing them into a slightly more optimal state. And because it's a better state to be in, then it's self-reinforcing.
Yeah.
And then that's why it continues to get better, right?
That is cool, right? It's so yeah, self-reinforcing in a sense that not only the network is sort of in this state, but also maybe the manifest behavior, whatever that is sort of like somehow more pleasurable or something. It's more rewarding.
Right. But also maybe, so if you think about this network as being, okay, so if you think about a particular network as being optimized for a particular task, and your network is somehow not optimized for that task, right? So for instance, my face memory network is not optimized for face memory. And but this assumes that there is a state that is optimal for face memory, right? And now say I push that network, my network, a little bit into the direction of the more optimal network. Then now when I see faces and I remember them a little bit better, then maybe it continues to push the network in the right direction, because there is a better network for remembering faces, for instance.
Yeah, that's cool. And so maybe from here we can talk a little bit about, I mean, and this is sort of an open question in terms of the mechanism of how this might work. I mean, actually, you had some really great paper questions in your TICS paper that are worth discussing as well. But one is sort of just the mechanisms. How nobody really knows exactly, but it seems stunning that networks at this detail can be enhanced or reduced. And not only, and the other question related to this is that you might change it during the session, but somehow that has an effect of changing the probability over time, over a long, changing your state such that that network is revisited more often in some sense, and that it's sort of a normal behaving network. But so what do you think? And this actually might tie into questions of consciousness or whatever, or it seems incredibly powerful that you have just a simple reward, you have this very specific network, and somehow they're reinforced and it's altered in some measurable way. Do you know what potentially the mechanism is? And is there some master controller in subcortical that does this?
So my guess is that, and this is actually some of Steve Gott's work as well, that the dimensionality of brain networks is much lower than we think.
Yeah.
Right. So it seems like there's an infinite number of states, and then if you reinforce, say, 30 times, and the brain doesn't know what you're reinforcing, it's just count, like saying, okay, this was good, and this was also good, and this was also good, and then you average out. But there are infinite states, so it's never going to average into something reliable, right? But if the dimensionality is much lower, and there actually aren't that many states, and whenever you reinforce a particular state, then there are a few different things going on within that time frame, and if you have enough time points, like enough reinforcements, and you average across them, then the only thing that remains is this one thing that you're actually reinforcing.
Yeah.
So, but for that to be true, then the dimension would have to be quite low.
Yeah,
which I think it is. So I think that's what's going on, that there aren't actually that many brain states that we cycle through in spontaneous activity. And then whenever you reinforce a particular brain state, if you have enough of these occurrences, then it just ends up reinforcing the one thing that is consistent across all the different brain states.
Yeah, so that's interesting. So in terms of behavior, so how many states do you think, I'm just out of curiosity, people, so it's interesting, probably people's intuitions range hugely on this. I mean, I can imagine. Right. I mean, the way you chop up states, I guess it makes a difference in terms of how you categorize them. But, right, it might be 20 or something on that order, I guess. But then how does that then relate to, I can't even imagine a state where, like back to the Shibata paper, that's a very specific, detailed activation pattern that I'm sure that there's millions of different types of activation patterns that are like that. And so,
but not in V1, I think, right? Because they were targeting V1.
Right,
so
right.
And V1 is very sensitive to these orientation gradings, right?
Yep.
So then to particular orientations. And we know actually from spontaneous activity studies in animals. So in cats, for instance, if you look at V1, you can see them cycle through these like different orientation patterns.
Okay.
So the fact that you find this activity for a particular orientation pattern in V1 actually isn't that surprising.
Okay, all right, that's interesting. And then it brings to the question of, so you go through these states, and they're either conscious states or not even conscious states, and so it brings back to another question, maybe even a bigger question, is like, what's the purpose of or a function of resting state in general?
Yeah, that's a huge question. I think that's one of the main questions I'm interested in, just in general. And my intuition is that it's just kind of a baseline for the brain. Like it's not that it has a purpose in and of itself. It's just kind of showing you everything that's happened before. It's like the cumulative history of your brain
Okay.
up to this point.
And it's sort of like it's sort of like the network's in this sort of ready stage, and it has to constantly be sort of, either spontaneously priming itself or something, or
yeah, it's like it's a measure of all your predispositions, right? So you have all the ways in which your brain is connected, like all the parts of your brain are connected, which is based on your history and like maybe genetics and whatever. And so this is your baseline, and that's what spontaneous activity is showing you. It's like maintaining this baseline.
Interesting. Okay. No, I think that's definitely, I don't disagree with that at all. Yeah, and it really is interesting in terms of the detail of the network. So that's one of the other questions too, is that when you choose something to reinforce, how far can you go in terms of can it be like, can I take you can imagine doing this for trying to help kids with ADHD or something, and so you find the whole attention network or something, that the entire network, or to reinforce. So I mean, or even how detailed can you go? Can you go down to layers? And that's another question.
Yeah. Okay, so there's a few questions here. So
yeah.
let's try. Okay, so first off, I think there are neural constraints on learning. There's actually a great paper by Byron Yu, or he's on it as well, I don't know if it's his paper, which is called The Neural Constraints of Learning. That they did a kind of brain-computer interface study in monkeys, and they were asking basically this, like, can you learn any pattern? And so what they did is they looked at the intrinsic manifold of activity in the monkey brain, right? So there are certain activity patterns that are more likely to occur and other activity patterns that almost never occur. And they show that a monkey can easily learn new activity as long as it's within the intrinsic manifold. So it can learn to shift activity. So for instance, say it's used to a particular activity pattern causing a forward movement of the hand, and they can easily learn that now it's causing a rightward movement of the hand. But the activity pattern is an activity pattern that exists. Whereas if you try and take an activity pattern that doesn't exist and give it new meaning, then the monkeys have a very, very hard time learning this. And we actually also saw this in the study we did with the autism, because we had a control group where we were trying to teach them a pattern that doesn't really exist in the brain, and they couldn't learn.
Interesting.
Right? So I think there is this kind of inherent constraint, which could be a hard constraint and it could be a softer constraint. It might be not that it's impossible for you to learn, just that it's much, much, much harder. So that's still an open question, but it definitely seems like there are things that are, so you can rewire the brain as long as you're respecting kind of this intrinsic connectivity.
Yes.
So that's one thing. And then the other question is, okay, so now with real-time fMRI, how far can we go? And I think that depends on your signal to a large, so how clean you can get your signal will be a big part of this. So if you want to look at more than 2 nodes in the network, for instance, and you want to look at entire networks, then it might be more difficult, because the more nodes you're adding, the noisier your signal is, and like the noisier your calculation, the feedback might not correspond to what you think you're trying to reinforce. Yeah. So I think it's a balance between cleaning the signal as much as possible and then reinforcing as much as possible. So I don't know. And layer connectivity is actually another really interesting thing that I'm very interested in.
Yeah.
So what we can do with layer connectivity so far is actually mostly with Vezo, right? That Renzo has been developing.
Yes.
And that's a very slow signal. That's even slower than the bold.
Right.
So I don't know if it'll work with real time. It might. So, and then that goes back to the first question of why is it working? Is it because it's a fixed delay, in which case maybe it doesn't matter if it's a longer delay, or is it because it's a slow signal and we're still riding on the edge of the signal, in which case if it's a longer delay, maybe it won't be possible anymore.
Yeah, and there's a chance that, I mean, maybe you don't have to, I mean, certainly with Renzo Huber's Vezo sequence that looks at vascular blood volume changes as opposed to capillaries. It's more localized to upper lower layers, but you may not need that necessarily.
You could do bold as well.
Yeah, you could do bold to, as long as you get something that's modulated on a layer, it might be good enough just to do bold. So in this case,
then it just depends like how clean you can get your signal, right?
Yeah.
Like if it's, because the real-time signal is way noisier than the post-processing signal we have.
Yeah.
So it's already noisy. And then you're adding on top of that another level of noise, and maybe it'll work and maybe it won't. I think it's just an empirical question. Yeah,
I mean, and one's hope is that the noisiness will be balanced maybe by the increased specificity in some sense in terms of what the signal means. Like, if you can get layers, you can get maybe potentially, some measure of cognitive control or feedback or output.
Yeah, I think the really exciting thing about layers is that you can then, I actually have a grant on this that wasn't funded, but I submitted. But I think it can give you a measure of feedback of directionality that you can't get any other way, right? Because if we have an hypothesis for what the directionality would be between different layers, then we can look at that even in the resolution of fMRI, where otherwise we're just seeing coactivations and we don't know what's happened before.
Yes.
But if you know that this is going from the deep layers to the superficial layers or vice versa, then you can start to imagine at least that you're looking at directionality.
Yeah, and that's huge, not only for understanding the brain and plugging into, network models or whatever, but also I think, right, for real-time feedback therapy in some sense, where you, it might be a more specific network that is more effective. So that's so cool. I mean, it's so exciting just to think about this, and right, we're doing everything we can to increase the signal to noise and going to high field strength and whatever. So maybe at this point, let's talk a little bit about the potential applications. So we talked a little bit about autism. We talked a little bit about looking at gradings, but then it seems like the applications are huge, and some are a little bit scary, maybe. But I mean, I can imagine it could be applied to like PTSD. It could be applied to any sort of application where you want to change someone's behavior, and potentially could even be applied to. I was just thinking about, well, let's say somebody has traumatic memories. You want to suppress those memories. You might be able to make it, find a network that's related to that, and then either reinforce it or not to help them in therapy in that sense. So you have more insight into this. You've been thinking about this longer than I have. So kind of curious your thoughts.
No, so I agree. I think the potential applications are boundless almost. I think the main bottleneck at the moment is that we don't know what to reinforce. We don't know what the networks are. So, and I think that's where neurofeedback also comes in, but at the more basic science level. So first we need to figure out what we want to reinforce, like which networks are actually correlated to a particular behavior. Yep. And the thing is, especially with psychiatric illnesses, the networks we know about are always very, very general, right? So like, okay, so amygdala is involved in okay, but amygdala is also involved in 5,000 different things. Yeah. So you can't just change amygdala.
Right.
So what exactly are you changing? Like what is the, and this goes back to the specificity question, and like how far can we push? But I think the first step is, and we're not there yet, right, for most of these things, is to really know what the networks are. Yeah. So and I think the way to do that is to have, I think the main thing we can do with this is using individual differences, which is something I've also been studying now for the past year and a half, so more actually. So we find individual differences in almost everything we look at, and they're actually really strong and also very reliable within an individual. And this goes back to spontaneous activity being a kind of baseline of your predispositions, right? And then you can use these individual differences in behavior to try and find the neural variance that predicts them. Right? So which neural variance predicts the behavioral variance, and then once you have that, then you have an hypothesis about which networks cause this behavior. You can then train them with neurofeedback, see if it actually changes the behavior you were measuring individually. Right?
Yeah.
And then you can move on to the next step of like, okay, so maybe this can be a clinical application.
Yeah, that's a really interesting angle on that. So sort of using the idea of individual differences to sort of enhance what's the causality of the networks that are related to that. Yeah, and I think that's right. It seems like the other problem that fMRI is struggling with is individual differences in terms of finding biomarkers. And so you average thousands of people, you find a biomarker, but because of individual differences, they're less effective than maybe they could be. And this sort of makes use of the individual differences in some sense,
Exactly.
which I think is really interesting in that regard. Yeah, and you can imagine sort of individual tailored sort of, you work someone up with a battery of tests, and then you find the networks. And yeah, and I guess you're right. I mean, there's so many, like attention is huge. It could be other things that you try to find enough of a network that is effective, but at the same time, you don't want too much to overlap with contradictory behaviors or opposing behaviors. And that's we're in early days in that, but yeah.
Because the level we understand these networks at the moment is very, very broad, right? So amygdala with DMPFC. Okay, but like so many processes involve this network. So I think we need to break it down a bit more. And also the other thing is that it could be lots of different networks. So for instance, if you think about face memory, so we have a paper with Alex where we showed that memory for faces is kind of subserved by a whole lots of different networks. Yes. Right? So it's not just face areas, and it's not just the connectivity of face areas to memory areas, and it's not just the connectivity of them to visual areas, but it's also to memory area, to social areas. And when you think about it, like of course that's the case, because of course face memory involves low-level vision and face processing and memory and social processing, because I know you are Peter, and I don't just recognize your face, I also know where I remember you from, and last conversation we had, and like all these other things I know about you. So it's very social.
Yes.
And if someone is bad at remembering faces, for instance, it could be a failure in any one of these different networks. And you could have two people who are equally bad at remembering faces, but actually for completely different reasons.
Yes.
Right?
Yes.
So it's not even enough to know what the face network is. You need to know for that individual where it went wrong.
Yeah, that's interesting. So yeah, no, I exactly. Yeah, as a matter of fact, it may not even be. It also brings to mind a little bit, the work by Jack Gallant, who comes up with these semantic spaces, and it seems that sort of any sort of, you can imagine a face area being sort of like that's where the faces are, but it's linked to, like you were saying, it could be linked everywhere to this massive semantic space. Yeah, that's right. And so how would you? Yeah. So I guess the question is, to the extent that it might not, and I don't even know with Jack Gallant's work that whether if he's actually studied individuals and how, I'm sure there's some order to it, but then there's some random, it's like a fingerprint in some sense, there's some randomness to it too as well. So it's also tricky. Okay. How would you go about, like, right, if you want them to help their memory, it would be hard, because you're saying, if the memories are, that network involved with that, you can imagine people have better memories because maybe they link it to more
right.
things. And how would you even begin to increase the network that does the linking?
Yeah. So I think, so we started looking at this actually specifically with face memory. And we found at least in typically developing controls, then it seems there are a few networks that seem to be most involved in predicting your ability to remember a face. And then I think what you might need to do is, for a particular individual, find out which of these networks is most important, like is most deficient in a sense, right? Like which network needs most strengthening. So going back to the ASD study, we also have some data on participants with ASD, where we saw that for them, it seems to be that the social network, like the link between the social network and the face memory network, is the one that's most predictive of their ability to remember faces, right? So that's where a lot of their deficits come from. Yeah. Whereas for TDs, it might be that, or it might be like a more memory link. And it could be different for different individuals. So once you know like the space of the networks, and you can try and figure out for a particular individual where they are.
Yeah.
And then you would target that network for them.
Okay. Yeah. I mean, right. It seems like it opens up an entire field of not only applications, but also, like you were implying, of actually understanding the causality of networks in the brain, and it's sort of a probe that can actually get at this more directly. Like you're saying, it's a little bit like external neuromodulation to get at causality, but here it's sort of internal reinforcement to get at causality, which is really cool. And it's amazing that it works so well. So why don't we go into a little bit in terms of just more general questions as we start to wrap up a little bit here, without going into too much detail. Is what's sort of like your ideal neurofeedback suite look like? What scanner would you use? What would you tell vendors to do? What would you add to it?
So we have Siemens scanners, and we have figured out a way to get around some of their packaging. So we can get the real-time data from the Siemens scanners, but it was a bit complicated. And I think it would be nice if the vendors allowed easier access to the real-time data, right? So that it would be easier for people to work with it, because there's really so much you can do with real-time data. Even just like AFNI's basic functionality of showing you subject movements during scans, right? Is so important that you can see your subject moving a lot, and you can stop the scan and say, okay, listen, are you uncomfortable? What can we do for you? Please stay still.
Exactly. And it's funny because go on. Sorry. You figured out a way around then that I would love to talk to you offline on how you did that. But anyway,
it was our physicist in the scanner who figured out how to do it. So
okay.
She did a great job. So now we have access to the real-time data. And of course, then you need a good interface. So AFNI does a great job with the real-time plugin. There are a few other options for using real-time, for getting at the real-time signal. You need to be able to reconstruct the images quicker. So especially with the 3T, it still works fine. With the 7T, this is becoming an issue. Right? It's like the image reconstruction isn't fast enough, and you start like accumulating delay as you scan, especially if you're doing like multi-band, multi-echo. Like as you get into more complex scans where each image is actually now 10 images. Yes. And as you go higher in the fields, then data is heavier and it takes a while to reconstruct. And that is already becoming a problem with the 7 Tesla.
Really? So 7 Tesla just because they're higher resolution images, I guess.
Yeah, and especially if you're doing multi-band.
Yeah.
Or multi-band, multi-echo, it gets heavy.
Is it like something that is solvable with like, a dedicated supercomputer or something like that? Or is it something?
Yes, I think it could be, but that goes back to the access problem. So Siemens does the reconstruction on their computer if you want, and we can access the data that they reconstructed. But taking the raw data and reconstructing it ourselves, that we haven't solved yet. Yeah. So then you can't have a special supercomputer, because that doesn't work, because you need to wait for the Siemens computer to reconstruct the data. Right.
And there's certainly, there's different groups out there with different solutions. Like we have our Gadgetron that sort of tries to take raw data off the scanner. And there's another company called not really a company, it's something called PulseSeq that not only allows you to pulse program sequences, but also take the data off before the reconstruction. So there's third parties. I don't know if the vendors like that much, but there's third party solutions that, and this is really important. I mean, this is it speaks to a different problem that's sort of ongoing too, is that it's great that we can rely on vendors, that make these scanners, because if they just relied on fMRI, we wouldn't have any scanners. It's all clinical, but then they only care about clinical. And so they don't think about optimizing things like this because it doesn't matter yet. But for them. But okay, so then going from there, let's say, then that's an issue. As far as the subject interface devices then. So is there like an optimal, do you think there would be an optimal type of feedback? Is it, you use visual, some auditory, tactile, other things like that? Smell could be interesting.
Smell could be interesting. We're doing a biofeedback study now with smell. See if, in sleep, to see if that works. So it's completely unrelated. But I think it would be interesting to try different kinds of feedbacks and see what works best. I think in the end, it will come back to what's most rewarding.
Yeah, you're right. You're right. Sort of like it could be or it could be a combination of visual and auditory or something like that. Sort of what, like a video game in some sense where it's immersive.
Yeah, I mean, if we could do a really cool immersive video game, I think that would be so much better than everything we've done so far.
Yeah. So then you have that, right? And who knows? I mean, maybe there's some sort of interplay with these signals with, for instance, heart rate variability or respiration. Or, it'd be nice to measure those and maybe get in sync with, sort of how, maybe use that also as a, sort of a signal of I don't know, how engaged the subject is or rewarded or whatever.
Yeah, no, I think that would be really interesting. I think what we really need to do for neurofeedback is maybe collaborate with animal researchers to figure out the mechanisms. So, for instance, one of the things I was thinking is we can measure prediction errors in animals, right, based on their dopamine neurons.
Yeah.
So it would be interesting to see at what point you stop getting prediction errors for the rewards. Like, do they learn at some point? Like, do they come to expect the reward after they create the brain activity? And that would show that they learned, right? That they know what they're doing, even though they don't consciously know. But if the brain knows that it's done something right, then now it should get a reward.
Yeah, that's actually right. I mean, I think that's a lot to learn, right, from the animal literature in that regard as well. It still brings me back to think about the mechanism of this and how does this dopamine signal sort of, how, you can imagine some sort of like, these subcortical structures that are involved with dopamine release, or it could be other, neurochemicals as well, but that actually reinforce these networks. I mean, it's to me, it works and it's so precise and it's something that I think we're just beginning to figure out. And if we can, it would be amazing. But yeah, so not to drift anymore, but is there so two other questions. So where would you, let's say this keeps on going. What's your hope, or what can you imagine the field could be like, let's say in 5, 10 or 20 years? I mean, what do you think the potential is for this? If everything goes well, if it sort of works at the beginning, and what can you imagine? Every hospital having this, or
yeah, no, I think the possibilities are really endless, because on the one hand, I said I think this needs to move into EEG for clinical applications, but if it really, really works and it's strong, then it could stay within fMRI, right? Because it would be, for a lot of these things that we could treat, it would still be worthwhile to bring them in for a few fMRI scans rather than lifetime of treatment. This could really work. So even though MRI is like impractical in the clinical setting, I think if it was strong enough, it would be worth it. So that's one direction, and also moving to other like more portable like EEG or whatever, and finding the right signature in the fMRI would obviously make things much easier. But I think the possibilities really are pretty boundless. I think if it works, then you can do this on. You just need to know what the network is. That's the big thing. It's like you need to find the right biomarker. And maybe this biomarker needs to be personalized for it to really work.
Yes. Yeah, because it seems like there's enough, like you were saying, individual subject variation that you can't just go in there and just assume that there's a certain specific network. And it might be tweaked. Like there'd be aspects of, I mean, you can imagine, networks, but also, different time scales and different, spatial scales of networks subnetworks that might be more specific, or who knows? I mean, it's wide open. Yeah.
Right. And like I was saying, it doesn't have to be networks or correlations or activations. Like if you could track in real time neuromodulators, you could feedback that, right? And that could also be a very promising direction clinically, or any other kind of signal that correlates with behavior.
So when you say tracking in real time neuromodulators, you mean like what? Like basal structures or neuromodulators like areas involved with release of dopamine, or what?
Yeah, like that. Like if you could track dopamine in real time, for instance.
Yeah.
Or if you could do MRS in real time.
Yes. Yeah, that's hard. That's really hard.
Yeah.
It's so much less sensitive, but it's not out of the question. And who knows? There might also be other markers related to dopamine release that could be sensitive to that in real time. Yeah, and I think that's the beauty of MRI, is that there might be, for instance, things like looking at neuromelanin, for instance, it's related directly to dopamine, areas where there's high concentrations of dopamine, and who knows, that might be sensitized to that, might be an indicator of this, of dopamine release. Who knows? There's a lot of opportunities. So is there?
We're counting on the MRI people.
Yeah. Well, we're keeping on looking.
Keeping on.
Yeah. So is there anything that I may have missed? Is there anything that you wanted to maybe mention that was worth mentioning, that I sort of glossed over or didn't? That's important, do you think?
I think we touched on most things. I think maybe the one thing that maybe wasn't clear enough was this distinction between spontaneous activity and kind of goal-directed activity. Yeah. So and this is one of the main differences, I think, between explicit feedback and the more implicit, covert kind of feedback, is that in the implicit scenario, then you're really just reinforcing these spontaneous brain states. Whereas in explicit feedback, you're trying to create the right brain state, right? Like you're trying to bring yourself into the right brain state, and then it's no longer really spontaneous. So I think that's one of the big differences between the two types of feedback. And I think, I think the implicit feedback has more potential for being effective. That's just my intuition. But also it's a really unique way of being able to study spontaneous activity, because like we said, we don't really know what these resting state networks are and what this resting state activity is. And almost everything you do would make it not spontaneous. Right? Yeah. So it would be difficult to study. And here you have a way of really directly modulating spontaneous activity and then trying to see what that does. So this is a way of answering this question about what is spontaneous activity, like what is its function, what is it doing? Yeah,
and if you're able to somehow modulate, like for instance, the number of the duty cycle or the time at which that state is achieved, over time, the dwell time, I guess, and that would be a good thing in terms of behaviorally. So it's sort of like, not only are you trying to enhance and strengthen that spontaneous occurrence of that state, but hopefully by doing that, you increase the amount of time going forward that state will be reached, as opposed to overt conditioning in some sense, or overt responses. It's sort of a mental strategy that you might learn.
Right, but then you have to actively engage in it all the time.
Right. You'd have to.
Whereas here, you're just like shifting the baseline.
Yeah, and it just works in that regard. And there's some people could argue that, well, it's good to have something that is like a mental strategy. It might be in some cases, but in general, I think you're right. I think that having like a state change that's would be more permanent, and you don't necessarily, you wouldn't have any sort of drift in mental strategy or whatever. It seems like it would be locked in. Bypassing the conscious process is both super potentially powerful, and some might be nervous by it, but I think it's actually it's all very it's above board and very clear what the purpose is, and I think that is I think it's really has a lot of potential, and it's an amazing thing that we can actually image the networks with high enough fidelity and with high enough resolution with fMRI that to be able to do this at all, and I think it has huge potential. So yeah, is there any and I think that and that is an important thing, because that's when I was thinking about even real-time fMRI or real-time feedback, even a week ago or so, I thought mostly was that, and then the other parts were the operant conditioning. But I think I'm really excited by this exactly what you're talking about. What you have been doing is that it does seem like it's a complete paradigm shift that makes it more effective and more elegant, and sort of ties into not only learning about the brain, but potentially very powerful clinical applications. So it's really exciting. I think it's one of the most exciting things that come along in a long time, and hopefully it'll catch on more.
I'm also really excited about this.
Yeah, I mean, hopefully there's more people doing this. I think, I don't think people are aware. I mean, it's certainly, that's one thing. You publish the papers and it's out there, but I think that people think it's cool and it could be useful, but I don't think they're truly aware of the potential of this in terms of tying into the spontaneous activity in this way. and bypassing conscious processes. So
yeah,
but
no, I really hope more people start doing this because it's such a large field. Like there's so much to study, and it's also very time consuming, which is I think one of the reasons people don't do it. Because if you need multiple scans for each participant, like in today's world with limited resources, it's complicated. But I really think that the potential is just huge.
yeah. Yeah, right. I really do. And yeah, it's just a matter of, getting it more refined and maybe standardized or whatever. So, one other thing I forgot to mention. So you're actually sort of part of the very beginnings of a pretty large institute being built. So do you want to talk a little bit about that? I mean, to what degree will real-time feedback play a role in that? What are you building at the Weizmann Institute?
Yeah, so the Weizmann Institute has one of the flagship projects this year, or like this decade, is building a new neuroscience institute, because we currently have, we have a few old buildings where people are clustered, and we have two departments, which is more like systems neuroscience and more molecular neuroscience. And the idea of the Neural Institute is to bring everyone together and create more interactions between the systems and the molecular people. And then you have like the molecular systems, then you have human neuroscience at the very edge of this. Right. So we're kind of at the tip of this interaction, but we are building a new fMRI facility as well. And we plan to, at the moment, we're planning 2 scanners, and the building will be ready in about 8 to 10 years. So, we've been thinking about what kind of scanners will exist in 8, 10 years. Like, what will we want? Will we want a 3 Tesla scanner, or will that be obsolete by then? Like, will we have 2 7 Tesla? Will we have a 7 Tesla and a 15 Tesla? Where are we going? And then hopefully this building will stand for some time. So, we want to prepare for the new scanners. Like, what will be the new scanners in 30 years?
Yeah.
What we'll be doing?
Yeah. Yeah, and so it's hard to it's hard to think ahead more than just a few years, but you want to hedge all your bets in terms of giving enough space in case you need it, or ways to integrate, and and the way neuroscience will evolve. It's hard to anticipate everything, but that's exciting.
I'm hoping that by the time it's ready, the neurofeedback will have really taken off, and everyone will want to do neurofeedback.
Yeah, and one can imagine. And that's the thing. I think that we're really close to, having something that's a clinical application that that other centers, not only clinical applications, certainly research applications are amazing, but for vendors, it really comes down to clinical applications, because that's what sort of drives the market for them. And having something like this that has a little bit of application, I think it would really cause the field to sort of take off if you can find something that sort of gets your foot in the door and will allow it to propagate more, have vendors pay more attention to it, and whatever. And this, I think, has a huge potential for that. So very last question. So what would be your advice for maybe a young researcher, like a undergraduate or even a graduate student getting into brain imaging? What's helped you? What would be some good general advice, maybe specific advice? Yeah.
I think the most important thing is to really know what you're trying to ask. Like, what is your question? And is the study you're designing, is it the optimal design for that question? And this also applies to neurofeedback, right? So are you doing neurofeedback because it's cool, or are you doing neurofeedback because it's really the best way to answer your question? And I think this is a pretty large pitfall of neurofeedback, because it seems like, oh, we'll do neurofeedback, it's cool.
Yeah.
But it really has to be tailored to the question that you're asking. So I think this is in general true of science or research. But so I think you have to be very, very clear about what you're trying to achieve, and then why you designed your experiment in this particular way in order to answer this particular question. And then, of course, you'll collect the data and you'll realize that it doesn't answer your question at all and answers a different question. And that's fine. That happens.
It happens often. Yes.
Yes. And it's great. But I think it's still helpful to start with a very clear idea of what you're trying to achieve.
That's that's really good advice. I totally agree with you. Yeah, the power of the technique, I think usually, or the power of the question, asking very clear, the better questions you have, the more the science will ratchet forward, I believe. And you're right, people fall into this trap of like, oh, it's just doing it. And I kind of want to see if this is possible and want to do this, which is good to some degree, but really, right, if you could ask an insightful question based on, what you know, to sort of, it focuses what you do a lot more, and it ratchets forward the field a lot better. That's really good advice. Okay, well, yeah, this was great. This is very exciting. And I'm hoping, actually, you've inspired me to start to talk to my own lab to maybe restart our real-time neurofeedback fMRI efforts towards these sort of questions and others. I'm excited. Yeah, and potentially we can collaborate. I'd definitely ask you advice.
Yeah, I love to collaborate. So.
All right. Well, thank you very much. Thanks for spending the time.
Thank you for having me.
All right, I appreciate it. And good luck in the future.
Bye, thank you.
Neurosalience is brought to you by the Organisation for Human Brain Mapping. This week's episode was produced by Anastasia Brovkin and Alfie Wine.